Okay, so to start out here, I'm going to talk about different levels of protein structure. So it's kind of different kind of degrees of understanding what a protein is. So think of it like you're a detective and you're trying to figure out what the protein is made out of. What does it look like? You probably already understand what it does and where, what tissue it came from because you had to extract it. This can be some very long and tedious work, by the way, that people do. I always like to tell the story of there was a graduate student at Oregon State University who joined a biochemistry lab, and his project involved working with a protein that was uncommon, but it was in heart tissue, so to get enough for his research, because he had to study it, so first you isolate the protein, then you decipher what it is. He had to work up a 55-gallon drum of pig hearts. And so, I don't know, they got them from a butcher somewhere or something. And so you have to do all these extraction techniques on 55 gallons of pig hearts. And it took him 6 months to go through the drum. And when he was done, he had about half of what he needed. So he was looking at doing another 6 months, another 55-gallon drum. And he said, forget it. Bye. I'm out of here. Like, that really happened. So he needed 3 nanograms of this protein for his study, and he had about 1.5 after going through an entire drum of pig hearts, which is not. No one gets into biochemistry to just be like throwing hearts in a blender and doing all this stuff. So it's tricky. So all the proteins that are real easy to get a large, quote unquote large quantity of, because remember nano means 10 to the minus 9, so like a billionth of a gram would be a nanogram. He needed 3 billionths of a gram of this stuff. But to get enough, if a protein is easily accessible, it's been studied. If you can get a bunch of it, people have worked it up already. So, it's getting pushed to there's something difficult. There's an obstacle that no one wanted to deal with right away. And now we're at the point where we have to deal with those obstacles. So those are kind of the proteins left to study. And the boundary is always getting pushed every year further and further. Now to start out here, besides obviously the fun and uplifting story of the pig heart blender of Corvallis, Oregon, we're going to kind of review just a little bit about the amino acid structure. So you've got, amino acid, you've got your amine group, your carboxylic acid group, so amino acid, you got an alpha carbon in the middle with a side chain, and so this is alanine, and it has a nonpolar side chain. And so, excuse me, these side chain interactions are going to be very important. So we talked about the properties they can have. This being nonpolar right here will be very important later on. And we can have ionized, unionized. Look, if you're looking at it, you're thinking that word's unionized. No. This right here, that is unionized. So it's always going to be the Zwitter ion form in physiological pH. So that's what you're working with. Just a reminder there. Some tables of the amino acids. I guess the one thing I'll point out, so there's proline right there. And so it's the only weird one. It has a little ring, but you don't have to do anything. So normally you see, NH3 CHCOO minus. That's what you're seeing for the backbone. This one has the NH2 and it has a weird ring. And it's the only one that does that. That's what they have the note here. So they call this secondary amino acid. That just means that there's 2 carbons bonded to the nitrogen, but I won't ever throw that one out in class. You'll only ever see the standard backbone. And so these are all nonpolar. These are all the common ones. We got the ones we can make through our own metabolic processes, and then which are non-essential. And then you have the essential amino acids, which we have to get from our diet because we can't make them ourselves. And we can make a polymerization occur through a condensation reaction, and we can repeat this over and over and over again. And this is how it's built in translation. Is it goes the end that ends in an amino group to the end that ends in a carboxyl group. So you're always adding on to the carboxyl groups. And so this is called the N-terminus. Well, I guess I'll show on the dipeptide. This is the N-terminus. Where do we get that creative name from? Well, there's an N there. And now if you had to guess, what do you think we call the other end? It's the C terminus. You might have gotten tricked and been like, oh, it's the O terminus. No, no, no. We go with the carbon. Okay, so you got the N terminus and the C terminus. All right, so that's just kind of that was a new idea, but in general, this is a review of what we talked about in the amino acid section. And so now we're going to start talking about, well, what happens when you get a long chain? So we call this a dipeptide. This is the beginning of a peptide chain, a polypeptide chain. Now what happens if I have 104 of them? And so cytochrome C is an enzyme. It's a very important, or it is a protein. It's a very important protein involved in cellular respiration, which we'll talk about at the end of the class. And here it is. Here's the chain. Does this order of amino acids? So every amino acid has a 3-letter abbreviation. So this is glycine, valine, glutamine, lysine. I think that's it. I can't remember. Aspartic acid. And so there's also a 1-letter version. If you ever take a biochemistry class, you have the joy of memorizing all of that. There's 23 of them or so. Now what we're looking at here is kind of the first level of understanding. So we have a protein here, and we know the order. We know the amino acids that are in it, and we know the order that they are connected. So it's like, I know I have a glycine and an aspartic acid and a valine and a glutamine and a lysine and then another glycine that's attached to 2 more lysines in a row, and it carries all the way through. And we have our N-terminal or N-terminus, so the one that has an amino at the end, and it carries through to our C-terminal, where we have a carboxyl group at the end. And so what we're looking at doesn't actually tell us as much as you might think about the protein. So we know how the chain is built. This is so far removed from actually knowing the structure of the protein, but it's generally where you start. So you get your protein, and then you denature it, which means you change its structure back. You at least affect the structure in a way where it doesn't function anymore. And in general, you denature it all the way back to being a chain. You throw an enzyme in, and it starts chopping off one amino acid at a time, and you analyze to see what that amino acid is. That's what you do. And so this is tricky. I'm making it sound a lot easier than it really is. I want to be clear about that. Like it's a lot of work. There's some amino acids get chopped off faster than others, and it takes a lot of repetition and comparing results to actually be able to figure this out. And the worst part is it still doesn't actually help you understand the structure of the protein all that much. So we call this the primary structure. So the primary structure of the protein is just knowing the order of the amino acids in the peptide chain. That's it. So you know what the chain is made out of and nothing more. Now, this chain gets built in translation. And when it pops off at the end, the whole chain is built, you still don't have the protein. You just have a long chain of nucleic acids that have been connected together with condensation reactions. It's a noodle. Most proteins are not noodles. They're blobs. So it has to actually kind of origami fold into a blob that will do its job. That was a great rhyme, by the way. I'm very proud of that. That process, again, we are as creative as ever with names. The process of going from noodle to folded protein, we call it protein folding. If you ever heard that term before. And it's an incredibly intricate process, and for every single protein, it goes differently. So it's like, to stick with the origami idea, you have a piece of paper, you want to fold it into a crane. Well, that's going to be different than folding it into, I don't actually know any other origami shapes. A paper airplane, because that's totally origami. It's a different process. They're different folds. You start with the same thing, you can end at 2 very different places. Now, I'm not aware of any proteins that actually have the exact same amino acid chain and then become different functional proteins that serve different purposes. But there are a lot of routes that any chain could go, and if it goes the wrong way, you don't get the correct protein. You just get something that has to get broken down for parts again. And so there's going to be 4 levels of protein structure we talk about, and this is the very first one. I just want to be clear that this does not tell us everything. That's all you got to understand right now. I'm talking about all kinds of high level stuff. We're not really going to go into that much detail in protein folding. You can look into it all you want. I'm going to talk about it, but I'm not going to really test on that idea. So the one thing I will say is the form equals function. So that the correctly folded protein, that idea goes hand in hand with the protein doing whatever job it's supposed to do. If it's not correctly folded, it's in all likelihood it's not going to do the job. So the last thing I'll say about protein folding is that in addition to just the fact that there's so many ways you could fold the paper, right? There's so many ways that this thing could start to interact with itself. There are also things called chaperonin proteins. So this chain might get built and then shoved into a chaperonin protein, and that will actually help it fold into the correct form. Oftentimes the chain is built and then pieces are snipped off midway through. All kinds of complex ideas come into play, and we're not going to worry about it too much, but it's at the forefront. Like, this is an idea that's at the forefront of modern research. I mean, protein folding is something that to this day is trying to be deciphered, and it has to be done separately for every single protein, because every single protein's folding process is different. We haven't found any sort of general trend that just, you solve one, you've solved them all. It's okay, we know how cytochrome C folds now, which maybe we do, maybe we don't. That doesn't mean that we're going to know how another protein folds. We're not going to know how collagen folds. This doesn't help us answer any other questions. And so once we know the primary structure, once we know the first piece of information, which is just what is the peptide chain that we're looking at for our protein, then we can start looking for secondary structure features. You might see a pattern in the naming here. There's 4, so it's going to go primary, secondary, then we'll have tertiary and quaternary. So now we're going to talk about secondary structures. And so this still doesn't tell us the whole idea of the protein. But what we can do is, once we know what the primary structure is, then we can come in and we can decipher it. So there are actually a couple things we can do with the primary structure. For example, we can break down, are there nonpolar, are there polar amino acids, where are they? Are they acidic or basic? So we've got this green, yellow, and red color coding here to help decipher with that. But we can also look at parts of the chain that are adjacent to each other. And those are going to give us these secondary structures. And what these secondary structures are common pieces. So proteins don't fold the same, but they often have little pieces that are repeated among different proteins. So one of them is an alpha helix. This is kind of like a girder that you might see in a protein, very common kind of girder form. And then there are beta sheets, often called beta pleated sheets. And then the third type is called a bend. It usually connects these beta sheets together. And we can actually figure these out because these are all parts of the chain. So these aren't distant parts of the peptide chain interacting with each other. These are neighbors. And so these structures are all held together with hydrogen bonds. So hydrogen bond interactions, attractive forces, are what hold these structures together. And these are like when you're building a house, generally all houses have walls and they have floors. And so that's what we have here. Is generally all proteins are going to have a beta sheet in them somewhere, or at least an alpha helix in all likelihood, probably a beta sheet, probably multiple alpha helices. And we can look at our primary structure and we can plug this into a computer, and the computer can tell us, like, oh, there's going to be an alpha helix here, there's going to be a beta sheet, throughout here, and then maybe there's like another alpha helix over here. I'm just guessing. I'm not actually circling things that would do that, just so you know. And so those structures that we see repeat among many different proteins, just common features, we call that secondary structure. So they're not the whole protein. So primary is the peptide chain for the whole protein. Secondary structures are just pieces within that protein. And we'll see it kind of in the big picture. Now, tertiary, this is the third level, that's when we know the whole enchilada, the whole folded form of the protein. And so now what you have is you have that chain and it has origami folded into a 3D structure, and distant parts of that chain are interacting with other distant parts. So when you have secondary structure, its neighboring areas interacting with each other. When you have the full tertiary, you know how the 51st amino acid in the chain is interacting with the 4051st amino acid in the chain, if at all. And you can have some interactions through hydrophobic, for example, if this is something that is surrounded by water, as we're seeing here, that means that hydrophobic side chains are going to kind of hide in the middle. Hydrophilic are going to be attracted to the water on the outside. And then we can have these ion-ion pairs. We can have a cation and an anion, acid-base pairing of side chains interacting to help hold it together. We can have hydrogen bonding, helping hold together things like alpha helices, beta pleated sheets, and we can have these disulfide bonds, or disulfide bridges, as it's labeled here, that actually have a covalent bond. And that's like, we are like stapling it together at that point. Like that's really going to hold it down. And so tertiary is when we know the whole structure of that folded peptide chain. And there's several interactions that we would know about. So if you ever see that there are a disulfide bridge occurring, or that there is a disulfide bridge occurring, or disulfide bonds, if you see hydrogen bond interactions, if you see ion-ion interactions, these are all going to help hold together the tertiary structure. They're going to help hold together the folded form of it. And so especially if you see this right here, if you see that, you know you're looking at a tertiary structure, at least. Maybe a quaternary. We'll talk about difference in a little bit. And then we have right here, these are generally how alpha helices are represented. They're just little kind of ribbons. So like this would be an alpha helix right here, this green section. It's just a coil. And then on the outside we just have, the peptide chain kind of folding around. And we'll talk about the details on this in the next video actually. I want to stick to just the levels of protein structure. So, quaternary. I've talked about the first three. So primary is just knowing the peptide chain. What amino acids are in it and what is the order that they are in the chain. Secondary are just pieces like alpha helices, beta pleated sheets. And then tertiary is like, okay, it's one peptide chain and we know how it's completely folded. We know its full 3D structure. You know everything about it. So what's left for quaternary? Well, I don't know if I've told you this already or not. I'm pretty confident I have. But proteins are complicated. I think I said protein folding was complicated earlier, and it is, but proteins are also complicated. So you can have something like hemoglobin that actually has 4 peptide chains that make it up. So quaternary structure only comes into play for proteins that have multiple peptide chains. So all proteins have a primary, secondary, and tertiary structure. But not all proteins have a quaternary structure. Because not all proteins are made out of multiple chains, but some are. And so if they are, then you have to know how those pieces fit together. So you can know, say we have, and to make things confusing, they really like alphas and betas. These are not beta sheets and alpha helices here. What this is these blue alphas, these right here. My square doesn't quite line up with theirs, but I hope you understand what I'm doing. That is a peptide chain that's folded, and this is the laziest, which means it's the most chemist representation of that folded structure, because we just drew I love it. We drew a square and we put an alpha in it. We're like, that's a protein. Okay, that's a peptide chain that's folded. And so what you have is you have 4 total peptide chains in hemoglobin, and there are 2 beta type peptide chains that fold into structures, and there are 2 alpha type peptide chains that fold into structures. And so hemoglobin is a protein that's used to basically tractor around oxygen in the bloodstream. And in the middle of it is actually an iron atom that's used to attract the oxygen and then release it when it's needed. And so to house that iron atom in the middle. We have 4 peptide chains around it. And once you know how all 4 of them fit together, so like this alpha right here, this has primary, secondary, tertiary structure. This beta over here, same deal, has a primary, secondary, tertiary structure. And then when you put them all together and you make hemoglobin, a whole hemoglobin protein, it has a quaternary structure because it has 1, 2, 3, 4 peptide chains in it. So if there's more than one, you have a quaternary structure. Okay, so to kind of lay it out a little bit more, primary are just the pieces, the order they're put together. Secondary are, can we kind of break that into bigger pieces? So we still don't know the whole picture, but we have some bigger pieces. And then when I'm going to zoom in on this. When we're looking at tertiary structure, oftentimes we have what's called a ribbon representation. That's what we're looking at here. And so we know the full folded structure of the peptide chain. And so when you see this little blue coil here, anything like that, that is an alpha helix. We got one big alpha helix here. There's a little one hiding behind it, kind of going left, right. So there's 2, and there's a third alpha helix behind a green beta pleated sheet in the middle. And so this would be a pretty common representation of protein. And so you're going to see these secondary pieces show up in proteins, and tertiary is knowing where they are, what they're doing, that type of thing. And this isn't always the case, but should you need multiple peptide chains, maybe 2 of these have to come together to actually form the protein, that's going to give you a quaternary structure. So that is what we're looking at. Okay. And then to kind of break it down in writing for you. So it's exactly what I was saying. You know, if you got an alpha helix or beta pleated sheet, those are secondary structures. They're not the whole protein, they're just pieces of the protein. They're some of the folding. And what's so special about them is they show up all over the place, and we can predict them from knowing the primary structure, from knowing the order of the amino acids. And the tertiary is, you know, the entire, you've got the whole enchilada. You know the whole story, at least for one peptide chain. And for a lot of proteins, that's it. You know the tertiary structure? Like cytochrome C, that's a 1 peptide chain protein. So that's just 1 chain of amino acids that folds to form that protein. You know tertiary structure, you know that whole protein. But something like hemoglobin, that's more complicated, can have a clustering of more peptide chains. And so they fold and then they fit together and form a larger protein that's made out of multiple peptide chains. And so once you know how those pieces are folded and fit together, you know the quaternary structure. Alright, and let's see, is there anything else I want to say here out of these notes? I'm kind of breaking it up a little different than I normally do. Let's see, we got a lot of protein activity. Okay, yeah, so I'm going to leave it there in this video. The last thing I'll say is when we're looking at tertiary quaternary structure, we've got, you know, acids and bases are going to attract because they're anions and cations. They'll repel each other if they're the same. Opposites still attract. Same charges will repel each other. We can get hydrogen bonding as well. So just depends on what we're dealing with. But these are all forces that help hold it. So that protein is going to get into its folded shape, and then it's got to stay that way. And so attractive forces are going to be what really work to hold it together. And so I will leave it there. And so in the next video, we'll talk about protein activity. We'll get into more detail on denaturation. And I think it might only be 2 videos I do for this section, 2 kind of longer videos. We'll see. Maybe there'll be a 3rd. I don't know. Oh, and then I don't know if I made this super clear earlier, but we were talking about, I think I did. So N-terminus, C-terminus. Yeah, I talked about that. Just kind of double checking my notes, making sure I didn't miss anything. All right, so yeah, so we'll talk about those other protein details in the next video.